If you've ever had a late night snack or been to the ice cream parlor, then your body has certainly employed the use of lipase, which is a critical enzyme in the digestion of lipids and fatty acids in the human body. And this will be the subject of my talk today. First, I want to step back and talk about some fundamentals. In organic matter, there are four major categories. There are carbohydrates, glucose, polysaccharides, monosaccharides, critical for energy consumption, lipids, which lipase facilitates the digestion of, is from a glycogen and three fatty acids. They form oils and waxes and plasma membranes in cells and organelles. We have nucleic acids, which help to encode the genetic DNA information for all necessary actions in the organism, as well as proteins, which are long strands of amino acids, and lipase firmly fits into this category. A little bit more on what an amino acid chain is. An amino acid is a carboxyl group, a central carbon, and an amino group, as well as what's called an R group. These two bond together in what's called dehydration synthesis to form a amino acid chain and an extra water molecule. The R group helps to give each amino acid a kind of special flavor by adding a series of compounds to give some interesting qualities such as a charge, a polarity, whether it's aliphatic or not, and the specific chains of amino acids, and given that there are 20 of them, can lead to a virtually unlimited variety of proteins to perform any action in the organism. Proteins are essential for transport. They are structural, they are hormonal, they provide signals for biofeedback loops, they help to transport oxygenated blood, they form the immunoglobulin and immune system response members. They are contractile in the sense that actin and myosin can help with motor function for muscle groups. And they are also digestive enzymes, which lipase fits firmly in this category to help process the various foods that organisms may ingest. So what is an enzyme? An enzyme is a catalyst. It helps to facilitate a specific reaction in a specific biochemical pathway in the organism. It lowers the required activation energy in order for a non-spontaneous reaction to occur, and based upon how many enzymes relative to the reactants that are in a particular area of a cell, they can control the rates of reaction, they can activate or inhibit these reactions, and there are cofactors and coenzymes which may more or less efficiently carry out the reaction that that enzyme is programmed for. These enzymes have a form and function. They have a substrate, which is a specific group of reactants that they will provide a perfect environment for in order to facilitate the reaction. There's an active site which acts as a lock for the key, which are the substrates, giving a perfect temperature, acidity, and orientation of the molecules in order to carry out the reaction, and they facilitate the reaction and eject the products accordingly. Every biochemical reaction has a specific enzyme which facilitates it perfectly, and that's why they are critical. They are the controlling function within the organism. So specifically, I have chosen gastric type lipase. It is a protein. It is an enzyme. It is found in Homo sapiens, that is humans, and its essential function is to digest short and medium fatty acids in the upper digestive tract. It is secreted by the actinar glands of the pancreas directly into the digestive system and will facilitate the first line of sort of digestive defense for those triglyceride lipids. The structure of lipase can be shown here. Its primary structure is 398 amino acids. This long chain is each letter represents a specific amino acid. There's 20 amino acids. There's 26 letters in the alphabet, so you'll see most of them here. So we start with methionine, tryptophan, and 3 sets of leucine, and that forms the specific polypeptide band that programs for lipase. The secondary structure is whether or not it is a helix or a pleated band, and where there are turns. Here's the general layout of the secondary structure. For example, the 29th to 32nd amino acids form a helix, etc. The tertiary three-dimensional structure can be shown on the left side of this slide, and that is formed when hydrogen bonds help to create a three-dimensional in-depth structure between non-adjacent amino acids. And these hydrogen bonds help to form like the real three-dimensional structure that forms like the active sites, and where cofactors and coenzymes can bond to these enzymes to increase or decrease their efficiency. And their lipase actually is a dimer in the sense that it has two lipase enzymes side by side in parallel, formed by hydrogen bonds in the 336 to 338th amino acid range in order to carry out their digestive function more efficiently. Lipase facilitates a specific biochemical pathway, and that is it breaks down triacylglycerol. So it takes a triacylglycerol, a water molecule, and produces a diacylglycerol, a free fatty acid, and a free proton. And lipase is the facilitator of this reaction. It is critical in lipid metabolism, and triacylglycerol produces a carboxylate group, takes in a water molecule. It has a coenzyme which is called hormone-specific lipase. It is creating free fatty acids as it continues to break down this triacylglycerol into diacylglycerol, and its eventual products are simple glycerol, fatty acids, carboxyl groups, and it's taken in water to perform that. It has some functional parameters, which are the ideal conditions through which lipase can function, and that is it works best when the acidity is 8.0, that is a little bit more basic, and the temperature is 37 centigrade, 98.6 F, which is perfect. That's what the homeostasis temperature of the human body is, so ideal. And I would like to talk a little bit about how lipase is programmed and where it comes from in the genetic code. In order to do that, I would like to give a crash course in the fundamentals of genetics. DNA, which is formed by nucleic acid base pairs and is wrapped around histone proteins in the chromosomes in the nucleus of every cell. Humans have 23 unique chromosomes, creating 46 pairs in a diploid cell, and the DNA will be transcribed by RNA, and that transcribed messenger RNA will mature and be read by a ribosome, which is made of rRNA and tRNA, to produce a long polypeptide chain. And there's a specific coding sequence on a specific chromosome which programs for the specific polypeptide chain which makes lipase. So to recap, we have chromosomal DNA, goes through the process of transcription, and is eventually translated outside of the nucleus to create the polypeptide chain, which lipase is derived from. The specific genetics of lipase can be shown on this slide. The gene that programs for lipase is called the LIPF gene, or more importantly, lipase F. It's located on the 10th chromosome, on the shorter leg in the 23.31 cytogenetic band. And if we look at a complete set of human chromosomes and we identify the 10th pair, it is on the shorter leg of that pair in the 23.31 cytogenetic band. And to zoom in on how that band kind of looks in a mathematical and diagram format, we can see the lipase operon here with some introns and exons, some coding and non-coding regions, and it exists kind of in series with a number of additional enzymes which may be programmed or maybe not, depending on the needs of the organism which is doing the coding. And to sort of conclude this talk, I would like to talk a little bit about the specific gene and protein sequence. Lipase F is made of 1,298 base pairs in this long convoluted chain. Here is the nitty-gritty series of nucleic acids which program for lipase. It's quite long, but it's all very necessary. And the amino acid sequence, which correlates very well to the UniProt reference, is shown here on the right. And this DNA sequence programs this amino acid sequence. The coding sequence is shown in this box on the left. It's made out of 1,098 coding pairs between number 65 and 1161. And that is how lipase is programmed. Here are my references. I leaned pretty hard on our open source textbook, Brenda, National Library of Medicine, and Uniprot. And thank you.